Hello friends, welcome to Botany option channel for UPSC examination. In today's video, we are going to see the plant anatomy in which we are going to see the TS, that is the transverse section of the dicot stem. In a very similar way, we are also going to see the TS of monocot stem, TS of monocot root, TS of dicot root, so that we can study the comparative anatomy of angiosperm, gymnosperms, and other plant forms as well. Not only the angiosperms, but we have to study the anatomy of gymnosperms as well, because UPSC straight away asks the question about the TS of ovule or TS of dicot stem or TS of monocot stem. Whatever it is, we have to cover all the plant anatomy section in the UPSC mains examination. All right. So let's get started. Now this is the unit number 3 in a paper 1, that is the Phanerogams, in which we have here the anatomy of C3 and C4 plants. That is, we have to study the anatomy of C3 plants as well as the C4 plants. So we have to cover both the monocotyledons as well as the dicotyledons. Alright. So we should take this anatomy part very seriously while preparing for the botany optional. Alright. So let's start with the transfer section of the dicot stem. This is an transfer section of dicot stem. From mains examination point of view, if the question come on the TS of the dicot stem, first you have to draw the diagram of TS of dicot stem, and then later you have to explain every part of this diagram. So first of all, we are going to clear our concept about the diagram. So the TS of dicot stem shows the three parts, that is three distinct parts can be observable in an TS of dicot stem. The very first is the epidermis, then we have cortex, and then we have stele, right? So the very first part is epidermis, second cortex, and third one is the stele. Let's begin with the epidermis. These epidermis are the single and the outermost layer. This particular layer is showing the epidermis of the dicot stem. Now, while writing down about the epidermis, you should mention that this is a single layer only. You can clearly observe this is a single layered structure and also has a thick covering above the layer, which we called it as a cuticle. As you know, the function of cuticle is to avoid the more water from the plant body. And then we have certain appendages on these arising from this epidermis. We called it as a trichomes. These are trichomes where the short and thick hairs on the stem, which are multicellular in structure. All right. So the very first layer is an epidermal layer. It is made of a single layer, and from above it is covered with the cuticles, and it has small projection called trichomes. All right. So this is the first part. The very beginning of the diagram, we have to draw the epidermis and then the trichomes above it. Then in the second part, we have cortex. Now this cortex present below the epidermis. That is, after the epidermis, immediately start the cortex. So this cortex is divided into three parts. First is the cholenchyma, then parenchyma, and then endodermis. That is, the cortex part is divided into three parts. So the very first three layers, you can see here, these are the spherical cells which really don't have intracellular spaces in between them, are compactly present, and these three layers are made up of the cholenchyma cells. Below that, we have a larger and circular cells with some intercellular spaces. These cells are parenchyma cells. And below that, we have one single layer of endodermis. So the cortex starts from below the epidermis. Then the first structure comes is the cholenchyma cells. Then we have parenchymatous cells, and then we have the endodermis. These three-layer cholenchymatous cells, also called as the hypodermis. These hypodermis are immediately present below the epidermis. All right. So the part of a cortex which is made up of the cholenchyma, it is called hypodermis. Then we have 3 to 4 layers of parenchyma with the intercellular spaces. That region also called as the general cortex. All right. And then we have the endodermis. So the cortex part is made up of hypodermis first, which is made up of cholenchymatous cells, then general cortex made up of the parenchymatous cells, and then the endodermis. All right. So you should remember carefully the second part, that is the cortex is made up of three parts. First is hypodermis, then general cortex, and then the endodermis. All right. Now, after the cortex, from here, that is from below the endodermis, all the rest of the portion comes under the stele. All right. So, rest of the portion of the diagram is comes under the stele. Now, stele is again divided into the various parts, namely the pericycle, then vascular bundles, and finally the pith. All right. So, the stele is divided into three parts. First is pericycle, then the vascular bundle, and then finally pith. The pericycle is present between the endodermis and vascular bundles. So, this portion comprising the pericycle, which is made up of the parenchymatous cells, and these cells are very compactly arranged and forms the hard part of the stem. All right. So pericycle just present below the endodermis and above the vascular bundle, and they are compactly arranged without any intercellular spaces between them. Then we have vascular bundles. The vascular bundles is made up of the phloem, then cambium, and the metaxylem. First, we have the phloem at the beginning of the vascular bundle. The vascular bundles are conjoint, collateral, and open. They were called conjoined because the xylem and phloem are present on the same radii, and that is why they are called conjoined. And between them, there is a presence of cambium ring. That is why they were called the open. So, in a dicot stem, we have conjoint, collateral, and open type of vascular bundles. So, in a vascular bundle, the very first part we have is a phloem. This particular part below the pericycle is a phloem. These phloems are made. This phloem is made up of sieve tubes, companion cell, phloem fibers, and phloem parenchyma. All right. Then after this, we have a second part, that is the cambium, which is often present between the xylem and phloem, and it is the region of rapid growth because secondary growth often starts with the phloem. All right. Below the phloem, we have xylem. This part is of xylem, and we called it metaxylem because here the tracheids and the vessels are very broad in nature. Actually, these metaxylems are the part of the primary xylem only, but the only difference is that it has a very broader tracheids and vessels, and that is why it is called the metaxylem. After the metaxylem, we have pith. The pith is made up of the parenchymatous cells, and the clear-cut intercellular spaces you can see here. The very large intercellular spaces are present between these cells, and this is the very central part of the stem. All right. So the last part, that is the stele, is made up of pericycle, then vascular bundle, and finally the pith. Here, in this particular diagram, you will observe this kind of structure at the very center of the cortex. These are called as the resin ducts. So while you are drawing or while you are writing about the dicot stem, you should remember it is divided into three parts. The very first is epidermis, then there is a cortex, and finally there is a stele. Epidermis are the single layered and have trichomes with cuticle. All right. Then cortex is made up of the hypodermis, general cortex, and endodermis. All right. After that, we have stele, which is made up of the pericycle, then vascular bundle, and finally the pith. The typical characteristics and the cells by which that particular region is made, you have to remember this carefully. Now, let us again see the various parts present in the tiers of dicot stem. We have talked about the first part, that is the epidermis, then the second part called cortex, and then there is a third part called stele. The epidermis, the epidermis are the single layered, and this single layer is from above it is covered with the cuticle. All right, and they shows the small projections. We called it as a multicellular epidermal hairs or say trichomes. Then second part we have cortex. The first three layers are made up of the parenchyma and we called it hypodermis. Then there are 3 to 4 layers of the parenchymatous cells and we called it as a general cortex. And finally we have here the endodermis, which is again a single layer. Then after this endodermis, the stele part started, where first we have the pericycle, then vascular bundle, and finally the pith. All right. So you have to draw these particular features in a TS of dicot stem, and you should explain accordingly the particular labeling in your diagram so that you will not miss a single mark in the question. All right. So this is all about the TS of Dicot stem. So if you find this video useful, please like the video. Please share this video with your friends who are studying the botany. And please subscribe to the Botany Option channel for the UPSC examination. Thank you very much for watching. See you in the next video.